It's 11 o'clock, so we'll get started. I want to thank everyone for joining us. Right now, there are already 34 people here, in addition to the three of us presenting. I know last week there was over 60 people who joined the Biological Sciences seminar series. So I'm expecting that more will come, and there was also a number of professors who joined us. So we're excited about this. We're really excited about this series, and we're really excited about having today Dr. Ileana Soto-Reyes. So before we get started, I just want to explain that those of you who are here, you can't see each other, you can't turn your camera on, but you can ask questions. So there is a Q&A button or a Q&A section. Please feel free to, as we go through, you can ask question at any time. Some of your questions may be answered, but that's okay. Feel free to ask your question. I will, Dr. Thomas and I will compile the questions, and there'll be a time for questions and answers afterwards. So we will ask your question to Dr. Soto-Reyes about her presentation. So the way we'll set it up is about 40 minutes. We'll have a presentation, and then we'll give probably 10 to 15 minutes of question and answer after that. And then Dr. Soto-Reyes will tell us a little bit about her path and how she decided to become a scientist and how she ended up here at Rowan University. So I know I won't steal too much of that, but I will, by way of introduction, let you guys know that Dr. Soto-Reyes got her bachelor's and PhD at the University of Puerto Rico, and then moved on to Johns Hopkins and the Jackson Laboratory. So I believe, Ileana, you do mouse research. I've done mouse research, and the Jackson Laboratory is something that anyone who's ever done mouse research knows about and has probably worked with. So that's really exciting. But we're excited to hear about your research and excited to hear about your journey here as well.
Yes, thank you so much for the invitation. I'm very excited to be here and to be able to share my research with our students, with Rowan, and our community. So I'm going to share the my screen. So can you see it well? Good. Okay. So I tend to speak very fast because I think maybe I have too much in my slides and I want to get, and I'm competing with the time. So today I'm going to try to don't talk too fast, but at the same time to do it on time. So let's see. Today I'm going to talk about how disruption of cerebellar postnatal development precedes neurodegeneration of Purkinje cells in a mouse model of Niemann-Pick Type C disease. And maybe this doesn't make so much sense now, but I'm going to talk about Niemann-Pick Type C disease. This is a genetic disease, and is autosomal recessive. What that means, if you took already genetics, is that the parents are actually carriers of the allele of that gene that is mutated. So they have a healthy version and they have the mutated version. But to develop the disease, you need to have both alleles, to be mutated. So they have the chance to have one child that is going to be unaffected. If they have 4, this is probabilities, not always is like that, but it's a probability that they would have one that would have both alleles healthy, 2 kids that maybe would be carriers of the mutation but will not develop the disease, and then 1 kid that it may develop the disease because, well, it will develop the disease if inherit both alleles that are mutated. This is also Niemann-Pick Type C disease is also a lysosomal storage disease. And what is that? What happens is that the protein that is mutated is MPC1, or it could be also MPC2. Both of them are actually involved in the trafficking of cholesterol out of the lysosomes and endosomes. So when cells do endocytosis, especially if they do endocytosis of cholesterol, it goes into late endosome. The late endosomes are going to fuse with the lysosomes, and then the lysosomes are going to be getting that cholesterol out of the lysosome through the MPC1 protein. So the MPC1 protein is in the membrane and it's the green one here, the protein, the green protein here. And then that cholesterol can go to the endoplasmic reticulum and Golgi complex to be then transported into the membrane where it's used. No? In MPC, because they don't have MPC1, as you see here, the cholesterol begins to accumulate inside the lysosomes. And then this makes the lysosomes to don't work properly. And if you remember maybe from your cell bio class and introductory class, or if you haven't taken that class yet about the lysosomes are like the stomach of the cell. They are going to digest, they are going to process even waste that need to be eliminated. So the lysosome is very important also for nutrition too. So if the lysosomes are not functional, then this can affect the function of the cells. And neurons are particularly very, very sensitive to this type of issues in the lysosomes. So in the case of MPC, one of the things that is going to happen is that neurons are going to die. There is neurodegeneration. I'm going to talk a little more about the disease before I go to the details, the cell bio details about the disease. But this is a disease that is also known as the childhood Alzheimer. And the problem with this disease is that children that have the disease, they apparently born healthy, and people cannot tell, the doctors cannot tell, their parents cannot tell that they are actually carrying this mutation and that they are going to develop such a horrible disease. No, this disease progresses with very early during childhood. So this is an example. These twins, before the disease onset, they were healthy. They were reaching the hallmarks of development, but then the disease the onset was actually after an infection. And then what is going to happen is that they are going to see developmental regression, cognitive impairment. So at the end, it's like having Alzheimer's. So that's why they call it childhood Alzheimer. Although it's a very rare disease, one case for 100,000 live births, I still, when I go and try to Google for getting more information, I still see stories that are, of course, very sad, because this disease is actually lethal. This disease is going to cause the death of these children, no? And their parents are actually knowing, once the disease is discovered, the parents are actually fighting against time and against a disease that is incurable, no? And trying to prevent the progression and also the death of their children. And sometimes even the math doesn't work, because sometimes in one family they got two, and even I have the opportunity to hear from a mom. She's a founder from a foundation for MPC. She had 4 kids, and out of those 4 kids, she lost 3 of them to this disease. And something that was very that caught my attention when she was talking, she was saying how her kids were born so healthy, and she was seeing them developing very normally and reaching their hallmarks even before it was expected. So there was no way to tell that they were having this terrible disease. And the 3 of them died very early, maybe teen years, and different ages too. So what happened in this disease? One of the things that I want to highlight here is that the presentation of the symptoms begin between middle and late childhood, and they include clumsiness, vertical gaze palsy, gait disturbances, and ataxia. And these particular symptoms are actually associated with the cerebellum. So here is the human brain, and the cerebellum is this one here, this brown region that you see here. The cerebellum has to do with coordination, balance, and that's why these symptoms that I mentioned are associated with the cerebellum. But the cerebellum also has some cognitive function. So it's even connected to the frontal cortex. So it has been even associated with ADHD, autism. So it has its own control, and functioning over the brain. Then we have the cerebrum. Eventually, the cells in NPC will begin to die in the cerebellum. That's why it's the first symptoms. But eventually, the cerebrum is going to be also affected. And this is the region that the sensory, the motor, everything is actually our minds is controlled in the cerebrum. So eventually, that's why they develop like a what we call a dementia. In the title, I think you saw that I'm working in a mouse model of the disease. So the reason why we study the mouse is because overall their brain is much simpler than ours. It's not exactly our brain, but it's very similar. And when we look at the cerebellum is exactly the same. And the neurons that die in the cerebellum in NPC are also present in the mouse, and they also die in the mouse model that I have. So that's why we use the mouse model, because it's actually recapitulating something important in this disease, that is the loss of these cells. So NPC deficiency, what happens? So this is the lysosome. NPC1, as I told you before, is going to transport cholesterol out of the lysosome. When we don't have NPC1, then the cholesterol will get accumulated. And what we know is that that is going to cause the accumulation of these lysosomes inside the neurons. And then that is going to eventually make these neurons to die. Obviously, the question here is that as an investigator that I have, why the neurons are dying, no? What is making the neurons to die in this disease, especially so early, no? Like in childhood. So I'm going to introduce you the cells. I told you the cerebellum is affected first. These are the cells that are actually dying in the cerebellum. They are neurons, they are called Purkinje cells. These Purkinje, if you haven't heard about neurons before, neurons have a special structure, and almost all the neurons have the same, in terms of structure. It's different, the morphology, but the structure is practically the same. They always will have a cell body, that is where the nucleus is, and they will have what we call a dendritic tree. And if you look, it looks like a tree, no, without leaves, but it looks like a tree. So this is the dendritic tree of the Purkinje cells. They also have an axon, but today I'm going to talk a lot about the dendritic tree. And you will see that I will be referring to the cell body versus the dendritic tree, because if you see, these cells is actually, they are arranged between each other. So here you actually can see the nucleus in red, the cell body green, and then the dendrites here are green. These are other cells that are in red in other regions, but this is the way that the Purkinje cells are arranged. They have that very complex dendritic tree. They are one of the cells with the most complex dendritic tree in the brain. But there is something special about them. They are actually flat. If you see them laterally, you will see that this dendritic tree is practically flat. And they are arranged like in rows like this. So this is a lateral view. And if you see, these cell bodies are here. There are two neurons, and there is the dendritic tree that is completely monoplanar. And then you see all the other trees in the back. So this picture that you see in the mirror is just the front, but if you look back, you will find other lines of Purkinje cells lining, no, and flat. Okay, then the other cell that I'm interested in and that I will be talking about today is the microglia. So neurons are not alone in the brain. There is a lot of other type of cells. Microglia is one of these types. They are the innate immune cell of the brain. That means that they are actually an immune cell, but that is living in the brain. And it's just found in the central nervous system. These microglia, they are actually cells that phagocytose. That means that they eat and they remove. They are going to remove, and they are in charge of clearance of dead bodies, dead cells, infections. If you get infection, they are going to remove the bacteria. So they phagocytose. This is their function, one of their functions. They are living surrounding the neurons. As I told you, neurons are not living alone. So here, these are neurons in other region of the brain, but you can see the microglia in cyan, this cyan color here. And they are contacting the neurons, and especially they are contacting also the dendrites. They have very specific functions in the brain, especially not only immunologically, but also they help the neurons to function in terms of synapses and in terms of connections. This is another image for you to show you the morphology. They are actually branched and they have very thin extensions, no? And they are very thin because they have to be making contact with the small synapses in the neurons, and they have very specific roles, especially during development and also in adulthood. So this is an example because microglia, it has roles during development, it has roles during the normal or healthy brain, but then they become like kind of an insult during disease. They become a problem. So this is an example in epilepsy. One of the roles of microglia in the brain is actually to help or to remove synapses. Synapses is the contact. This is red is one neuron. This is a contact from one neuron with this yellow neuron, no? So these are the contacts. These are the synapses between neurons. And microglia is always surveillance or checking now if those synapses are working properly. If they are not working properly or they need to be eliminated, which is something that happened normally in the brain, especially during development, microglia, because it's a phagocytic cell, is going to remove it. But if the synapse is working okay and is necessary there, the microglia is not going to remove it. What happened during disease is that microglia become activated, and I will show you examples of activated when I talk later on, become reactive. It's like becoming angry for something that we sometimes don't know, but and then it began to remove synapses that are actually needed. And if these synapses are removed, for example, in this case, it's removing a synapse that is actually inhibitory. That means that it will make the electrical activity of that neurons to decrease. If you see, this is the normal electrical activity, but when that inhibiting synapse is removed, then the activity is increased, no? And that's when people have seizures. That's an example here. The same, it can remove also other synapses that are actually necessary for the normal function of the neurons. And at the end, it's going to promote dysfunction of these neurons. And obviously, it's going to also contribute to disease. We still, in different diseases, we have to understand why microglia will target certain synapses and why it does that, or why it's activated. So that's part of the research. But today, I'm going to talk about how microglia is acting in the disease that I'm studying, no? And what are my questions about it? So here we come back to my drawing, showing you that the accumulation of this lysosome is going to cause the degeneration of Purkinje cells. But also, my question is if microglia is actually contributing to the acceleration of this degeneration. And the reason why I'm asking that question is because microglia get activated during degeneration and it's going to become a killer. And also, in this case, because this is a genetic disease, all the cells have the same mutation. So in this disease, microglia is also accumulating phagosomes. It's also accumulating lysosomes. So we call it phagosomes because it's from what it has been phagocytosed. So imagine this is a cell that is eating a lot. So it has to use the lysosome to digest everything that is eating or phagocytosing. So if it's having these issues with not having MPC, this cell is also going to be accumulating these phagosomes. And therefore, we expect that the microglia is going to be also dysfunctional because it also has the disease, no? So how this diseased microglia is going to affect the diseased neuron, no? So this is the question here. So that was the first experiments that I did. I'm going to show you the image. Here, I'm going to show you the phagosomes in red. I use a lot of microscopy, so I'm labeling structures and cells. using antibodies that are fluorescently labeled. And this led me to see what I want to see. No, I have to buy what I want to see. So this is first I have to know what I want to see and then buy the antibodies. But so I'm going to be using the MPC one NMF 164 mouse model. They actually, in this case, is a point mutation. The MPC1 protein is misfolded and then is eliminated, and that's why they don't have MPC1. They don't have MPC1. So they develop the disease. And once again, microglia is the phagocytic immune cell of the CNS. So this was my first staining to see, to explore the mouse for the first time. And then I found this marker was in green is labeling the cell bodies, the cell soma of the Purkinje cells. This is myelin. Myelin is actually in the axon. I was just checking to see if something was changing, but then I labeled the phagosomes in red. And if you see now, this is a 10 weeks mouse, you see all these cells, no, like very round cells that has red staining, and you don't see them in the control, no? So this means that these are cells that are actually accumulating the phagosomes in their bodies. And then if you see too, there is no green bodies here. They have disappeared because this is the degeneration of the Purkinje cells. So you can tell from this image that the neurons are degenerated and that probably microglia has eaten everything there, no? Like they have been completely, and that's why this microglia is suffering on digestion. So this is another staining. In this case, I'm staining the neurons and the dendrites. And something that I found very, the first time that I'm doing this, I was like very surprised that we actually see this accumulation of these microglia in regions where the dendrites are completely gone. We can actually see sometimes the cell bodies. So it was like they were eating the dendrites first, even before the cell bodies. So that kind of caught my attention. I was like, wow, why the dendrites are so yummy, no? And myelin tends to be very immunogenic, and it tends to be very attractive to be phagocytosed, but we don't see that many microglia activated in that region. But the dendrites, it seems like, oh my God, like the microglia is really onto them. So this is going to be practically my talk today. Investigated that. So first, I wanted to establish if we are studying a neurodegenerative disease, by the time that the neurons are all degenerated, nothing can be done. So we want to investigate what happened first. What is preceding the death of these neurons, no? Because we want to fix that problem and then prevent the death of the cells, no? So I was working in this mouse for the first time, so I wanted to then identify what is early, what is the early stage in MPC, NMF 164 mouse model. For the first thing that I thought, it was like, okay, let's look for the time where the neurons are not degenerated yet. So when we look, I did 4 weeks, 8 weeks, and 12 weeks. Obviously, 8 weeks and 12 weeks, we have a lot of loss of neurons. The neurons are here, these cell bodies that are big, practically they are gone at 12 weeks, and 8 weeks is like almost a 50%. But then when we look at 4 weeks, there was no differences between the wild type and the MPC mouse. So then I said, okay, 4 weeks is our early stage. So now I'm going to look into microglia. What happened with, before that, we actually look also in terms of function, no? I told you that if they lost the neurons, they are going to have ataxia and they lose coordination. We did this test, that is the horizontal ladder. We measure the misses and the sleeps, how many times the mouse was sleeping in between the runs. And if you see, it's very interesting because at 4 weeks and 8 weeks, we don't see differences. And then the differences are actually very severe at 12 weeks. And this is actually what happened in neurodegenerative diseases. People don't have symptoms until they have lost a lot of neurons. And by that time, it's too late. Nothing can be done, no? So, and usually those stages where are still loss of neurons, may not see anything, and that's why it's like a very abrupt change, no? And when they go and check the brain, it's too late. So this is an issue that we have in neurodegenerative diseases. Okay, now let's look at microglia. What happened at microglia? And before I go that, this is the wild type 4 weeks. This is very young. The microglia cell body is stained in cyan blue, and then the phagocytes once again are in red. This is a wild type. You see that they have very little phagocytes. Sometimes you can see a little more at 4 weeks wild type. This is the molecular layer where we have the dendrites of the Purkinje cells. The Purkinje cells in here, this is including, it's the same picture, but including the dendrites and the Purkinje cell bodies, no? So when we look at the other stages, obviously at 12 weeks MPC that we have lost the majority of these Purkinje cells, you see that those cells are very wild, no, very big, swelled, full of phagocytes, and they are even losing their morphology, their normal morphology. At 8 weeks is the same because 50% of the neurons are actually lost. But then when we look at the 4 weeks MPC, it was very interesting because at this stage, we are still having not death of Purkinje cells, but if you see that region of the dendrite is full of these microglia that look like not only they are activated, but they are also accumulating these phagocytes like they have been eating for a long time. And we are assuming that there is no degeneration here. So why they are so full at this early stage? And their morphology is very different to the 4 weeks wild type. We also quantify the number of cells, and we found that there is definitely a significant increase in the number of these cells. So that means that they were also proliferating. And this is part of the activation of microglia to proliferate during degenerative situations, no? So we also look at the morphology because in microglia, the morphology tells us a lot. So it's part of their activation. They are going to retract their processes, and they are going to be thicker and fatty and larger. So when we look, we definitely see that at 4 weeks, MPC, they have a larger volume than the wild type, and their processes were also thicker. They were also thicker than the wild type. Sorry, I don't want to. Okay, then, and if you see this accumulation of phagocytes, we did those quantifications too. I don't have the graphs here, but they are very obvious, no? You see very small phagocytes in the wild type, and there is a huge accumulation through the cell of these phagocytes. So they are in the molecular layer where the dendrites are, and we are thinking, okay, and they are active, no? Because they are so angry and they look so activated. So are they actually maybe interacting with the dendrites that are there, the dendrites of the Purkinje cells? So we measure this with a special software. These images are actually 3D, so that's why you see the lateral view here. It looks like a sandwich, no? Like in the middle of the flat cells, the microglia are there. And we actually had the opportunity to quantify how much this microglia was actually contacting or maybe engulfing these green structures. So in red is what we quantified as regions that were contacted by the blue, no, the cyan microglia label, the labeled microglia. So we definitely found that there was more contact of these microglia, or maybe more engulfing too, of these microglia doing with the dendrites, no? So they were actually, we thought like, okay, so at this early stage, they are already contacting these dendrites, so they are eating the dendrites and they are promoting the degeneration. And when you look at 12 weeks, it's even obviously worse, no? This is the time where the degeneration is actually practically done. But then you can see this massive loss of dendrites. You still can see some cell bodies. So, and you see those microglia, no, like over that degenerating neuron, like this one. Look, this dendritic tree is completely like all death, no, and the microglia seems like they are like chicken, like eating it, no, over it. So this actually remind me like a lion when they get the meat and they are trying to, that's exactly how I in my mind, I see this microglia getting into the dendrites. And we can think, okay, the neurons are degenerating, but why they are targeting the dendrites? Are they actually killing the cell faster because they are eating the dendrites? So imagine if you are sick and somebody begins to cut your arms, your legs, and then you are bleeding. So obviously you are going to die faster, no? So we decided to challenge the system and see if we could see more degeneration of dendrites when we overactivate microglia. So we use Western diet. Google said, no? Like we did when I was in the Howell lab before coming to Rowan, we did this study in mice, and we give these mice this diet that was designed very similarly to our Western diet. And if you see, it has high fructose corn syrup and sucrose that is actually absent in the control diet. We also they have much more fat than the control diet, and the protein was mainly from animal. And this control diet in the mice is very vegetarian, so it was a Western diet for these mice. And we saw in this study, we knew that microglia was getting activated by this diet, so I decided to use it in our mice. They began to take to be fed with the diet when they were 21 days, and we did the treatment for 5 weeks. So they were 8 weeks when I sacrificed them. So this is at 8 weeks. This is the regular diet, MPC1. Obviously, there is activation of microglia at this time because there is loss. But when you look at the Western diet, the activation of microglia, the inflammation is much more, no? They are much more aggressive, and there is much more proliferation. So now I wanted to look at the dendrites and see what happened if they were eating more dendrites. And it definitely happened that yes, when we do the quantification, there was a significant loss of dendrite that compared with the wild type, and there was no differences in terms of the MPC regular diet and the wild type, but it was significant between the Western diet and the wild type. So if you look at here in this image, you will see these holes that doesn't have dendrites, and you will see that the microglia is actually there, no? And it's actually already also over the other dendrites. But the other thing that we found interesting is to find also cell bodies that were by itself, no? Like the dendrites were eaten first. So with this study, we actually conclude that at very early stage, 4 weeks, MPC microglia is already activated and is already contacting and maybe engulfing dendrites at this early stage. It's all obviously it becomes more, it becomes worse at between 8 and 12 weeks. And Western diet amplified that degeneration of the dendrites and that phagocytosis of the dendrites by the microglia. Now, the most interesting thing here was to find that the microglia was already activated before the death of the Purkinje cells. And the question was, this was my early, so now it's like, okay, we have to go earlier because something is going on with these microglia. They are already activated. Why? Not before the neurons die. So if you look 4 weeks, this is the end in the mouse in terms of the lifespan of the mouse. This is the end of development. No, that's they are an adolescent at 30 days. So if we want to look before that, we have to go to postnatal stages of development, and that's how we then went there. No, like we said, like, okay, we have to begin to study development, and I'm glad that we are, because it's very interesting. So in contrast to the rest of the brain, the cerebellum is actually mostly developed through postnatal development, after birth, no? And this is how it begins, and then you see how the structure is getting more growing and getting the structure that is supposed to be the lobes and also the regions of the cerebellum. I don't have time to go through, but I just want to tell you that I want to my next question was, how does MPC1 deficiency alter cerebellar postnatal development? And remember, even the mouse, in the case of these mice, the patients, they born apparently healthy. So what is going on during development? Because obviously something has to happen so that the neurons die so fast, no? That is actually kind of silent, that we don't see it in terms of symptoms. So my hypothesis, it was that MPC1 is going to...
Quick reminder that we're at 30 minutes.
Oh, okay. Oh my God. So MPC1 is going to cause the dysfunction of lysosomes, and this dysfunction can affect microglial cells in terms of their immunogenic response and phagocytosis, and they also can cause, this dysfunction actually affect the development of Purkinje cells in terms of dendritic growth and patterning, and also synaptogenesis and remodeling. So this is the microglia at P4 in the cerebellum. They are actually, they are not differentiated like in other regions of the brain. They are more round, and they are not having their processes. They are also accumulated in the cerebellar medulla, where they migrate. They are migrated into the other regions of the cerebellum. What we found here, it was that the migration, we found less cells closer to the cortex, so the migration of these cells was actually affected at this early stage, P4, very early. Then we look at the proliferation in those cells, and we see that they are proliferating more. So we use this marker, Ki67 is in red, is a marker or proliferating neurons, so proliferating microglia. So the cells that were green and red, so they will look yellow here, they are actually cells that are proliferating, and we found many more in MPC. Therefore, when we look at P10, this was P4, when we look at P10, and this is a wild type versus an MPC, then you see that there is a lot of microglia. This is in green, microglia is in green, in the MPC cerebellum. So at these early stages, we already see proliferation, a lot of proliferation of microglia. When we look also at the level of where the Purkinje cells are, we see also a lot of these microglia accumulating in that region because there is more proliferation. And if you see also the morphology is very different, no? They look very already like activated. And at this stage, there is no cell death or anything. This is just development. And then when we look at 14 days, we still see many more microglia in the cerebellum of MPC mice, but we also found that they were having these structures that are round. Those structures are actually phagocytic cups. That means that they are actually containers, that they are actually getting pycnotic bodies, apoptotic cells, and they are getting there like engulfed. And then if you see here, this microglia has two phagocytic cups, and inside of those phagocytic cups, they have nuclei, because those are apoptotic bodies. Those are pycnotic bodies. Those are cells that are dead, and this is a role that microglia has during development, to remove cells that are dying, and this is normal during development. But in the MPC, we found that these microglia were having more pycnotic bodies and also more phagocytic cups. So that means that they were phagocytosing more cells or more apoptotic cells. So in MPC, proliferation is affected, but also the phagocytic activity of this microglia is increased. We also have, and I hope I don't want to take too much time on this, but we have the climbing fiber refinement. These climbing fibers, you see one blue and one green, are actually the presynaptic terminals that are going to interact with the neuron. They begin first interacting with the cell body, but eventually one of them is going to be eliminated, and the winner is going to be getting translocated into the dendrites to make contacts to and make synapses with the dendritic tree. So we measured that. You will see the contacts in between the climbing fiber with the Purkinje cells in red in the picture. So those are, you are just seeing the contacts, and that's why it looks like a grain. So what we saw is that these the translocation was obviously normal in wild type. You see the red bGLUT2 ponta or the input in the dendritic tree, and there is less in the cell bodies of the cells. But then in MPC, we see that these inputs are actually more in the cell body and much less in the dendrites. So the translocation is not working properly, and they also have much less of these inputs. So, and then if you look here too, this is the quantification, we saw that the microglia was actually interacting more with the cell bodies in this case. And it was like they were trying to remove those synaptic contacts that were in the cell body that were not supposed to be there. We quantify how much of those synaptic contacts were inside the microglia, and we found that those ones were increased in MPC. So this microglia is eating more of these bGLUT2 or these presynaptic terminals. This is just a video. The two first ones, these two, are actually MPCs, and this is a wild type. And all the question is there anything that we can see that maybe is not normal in any behavior that is affected in the brain? This is or in the cerebellum. This is 4 weeks, and we quantify that these mice are actually digging more. The behavior of digging is actually normal in the mice, but when we look at the MPC ones, they are actually having an increase in the digging behavior, and they also do that for a longer time. So this is, then we said, okay, this looks like a perseverative and repetitive behavior, which is almost always associated with autism, for example. And the cerebellum has been associated with that behavior too. I don't know if you see now the wild type is doing it, but these two, I think at the end is where this one is doing it, and yeah, they tend to do it more. And here they are doing more. Okay, then finally, this is my conclusions. So in cerebellar MPC1 deficiency in cerebellar postnatal development, specifically in the microglia, we see that the migration is delayed, the proliferation is increased, and the phagocytosis is increased. In terms of the Purkinje cell climbing fiber synapses, we see that the density and the translocation is actually decreased, and the phagocytosis seems to be increased of these synapses. So we published this paper. This actually, all this work that I have presented in the other paper and this have been students from undergrad students from Rowan, and I'm very proud of them. They have done such a nice job, so they have worked very hard. This is the ongoing project. We are studying what happened with Purkinje cells in terms of their development of their dendritic tree, that it happened during postnatal stages. We see that, surprisingly, that in MPC, the dendritic tree is larger in terms of branches. This is at 14 days. But then at P30 is decreased. So something is happening in between P14 and P30 that's lost. We also have mice that are labeled with GFP, and the Purkinje cells are expressing GFP, and we want to see if the microglia is actually eating more of these dendrites before the 30 days, and that's why we have the decrease. And we have some preliminary data that suggests that yes, that this microglia is actually phagocytosing more of these dendrites. So we still need more information. This is ongoing. This is just preliminary. And this is acknowledgement. Obviously, this work could not be done by myself or anything. So I have my students. They have worked so hard, and they have been so dedicated. All those quantifications, all those graphs are students doing these quantifications. I have my collaborators too. Actually, right now with Dr. Caron, we are actually going to do, we are in the process to do analysis of RNA-seq to see what genes are changing. And then this is the fun part, no? When my students can present their work and when we can be together. And I miss them so much because we haven't been able to do that anymore. Okay, so questions. I'm ready to answer any questions. Did I do a good time?
You did perfect. Thank you so much, Dr. Soto-Reyes.
You're welcome.
Actually, first, I want to ask a couple questions that came in kind of about basic research. Okay. You're using this beautiful mouse model. Can you tell me how exactly do the mice become NPC1 deficient for them to be used for research?
This is very interesting. This particular mouse was actually an spontaneous mutation that was discovered in the Jackson labs. They are always checking the mice in terms of how they behave, and they saw that these mice, they were one of them probably born with this developing ataxia so early. So when they check and they went and they go to the genome, they discovered that they have this point mutation. And it's not a human mutation, but it's very similar to one of them. It's close, but it develops the same disease because this is actually in the MPC1 gene, and then, yeah, and it causes the deficiency of the MPC1.
That's awesome. So usually you will design a mouse model, but this one was actually found.
Yeah, exactly. And this happened a lot in the Jackson lab. Because they are producing so many mice to sell them, no? They also monitor the mice, and then there was this spontaneous mutation, and then they did all what it need to be done to then discover which was the gene that was. But yeah, and there are other models. They sell models that are actually with one of them. There is one that they actually introduced the human mutation. Is a transgene in this case. But I began to use this one because I was in the Jackson lab. One of the collaborators or one of the investigators that had them. And I went to him and I'm like, I want to use this mouse. And he's like, yeah, get it. And he gave it to me for free. So I was like, oh my God. And I was so happy with that. Yeah. So that's why I haven't checked yet.
So you have all of these gorgeous pictures. Can you tell us a little bit about the process involved in taking the pictures and showing these neurons so well?
Yeah, so we have a fluorescent microscope when we have to do very simple quantifications like the cell bodies and that. But to do the dendritic tree and to do the contacts if the microglia is contacting, we have to use confocal microscope. And the confocal microscope, it actually have that capacity to do 3D imaging. So we can have those 3D imaging. It actually take a lot of time because it has to scan through the tissue. And each picture take like 5 or 7 minutes, and it's like, oh. So yeah, but it's great what and I have the software, the IMARI software, to be able to do the quantifications. Because one thing is to get the image, but the analysis is actually a software. It cannot be done if you don't have the software. Yeah.
Another question. Do the microglia also eventually consume the cell body of those Purkinje cells, or just the dendrites?
No, they seems to also get the cell bodies too. Because if you see, there is loss of, sometimes you see whole loss of everything, no? Like we focus because it was interesting to see those cell bodies randomly getting there, no? Like, and it was like, okay, so it seems that they are eating the dendrites first and maybe the cell bodies later, but they will eat they would remove the cell body too.
So would you consider this a type of autoimmune disorder?
Not so far, because for an autoimmune disorder, we will need antibodies, no? And it definitely, in very later stages, people have found in neurodegeneration that the adaptive cells, no, of the immune system, they will invade the brain, because a lot of these microglia will release a lot of cytokines and chemokines, so it's going to attract cells from the immune system. But because the first innate stage, no, where microglia is acting, and there is other type of cell too, astrocytes, that are involved there too, it's not involving antibodies and T cells. We would not say that it's autoimmune.
So I know you said that the mice lose coordination by 12 weeks, but do they show any other symptoms similar to the symptoms in human besides loss of coordination?
That's the thing, yeah, we cannot measure the eye. One of the main one is actually the eye movement, no? Like the kids, lost the vertical movement of the eye. We cannot measure that. Definitely the ataxia is very, very obvious, and it's the one that we can measure. But early on, if you see they are smaller, the mice are smaller. I don't know why, but they are smaller. And the only thing we got was that activity of digging more. They are more hyper, I think. So we need to do a little more to study what that hyper means. But we actually like the digging thing because it looks like it's a repetitive behavior, which is, it has been linked to the cerebellum before in models of autism.
And do they show seizure activity at all?
No, they don't get seizures. Yeah, that's something that's strange because patients, they tend to have seizures, but the other thing that I think these mice don't develop, they don't have the loss in the cerebrum, so the loss of neurons in the cerebrum. I don't know if it's because they die too early, but mice are tricky. Even Alzheimer's disease, I work in models of Alzheimer's disease before, and they don't mimic the death of cells like in the human, especially in the cerebrum. So that's why I also select these mice, because I was like, this one have cell death, and that's what we need to study, no? But yeah, maybe that's why they don't have the seizure, because they are not having loss in the cerebrum.
And do you notice a macroscopic architectural change in the cerebellum, like a smoothing of the cerebellum too? I don't know if you said that or not.
It gets smaller. It gets reduced
Okay.
in size. Yeah, definitely. Yeah.
Another question. Could the results discovered in mice regarding the Western diet also be applied to humans, do you think? And does this status suggest the Western diet contributes faster destruction of these neurons, or?
Yeah, I think if of course that requires much more deep. We didn't claim anything in that paper, like saying, oh, Western diet, or we didn't even put it in the title or anything, but because we use it as a tool. Yeah, because you always need more, no? Like to be able to translate that into humans. Obviously, the humans are disease is much more complex, and we have more complications. Like, for example, the kids are developing this disease sometimes as the onset happen after an infection, no? And like, so there are other aspects that have to be studied, but definitely it's not a secret that Western diet is not good, no? And it has the question if it's that, if there is, the reason is because we don't know if it was a rise in glucose, no? That then that would make the microglia maybe get more activated, or like, we don't have the answer the why, but we know that it definitely glial cells, especially in cases where there is already an insult, this the Western diet become an insult too, no? And the mice tend to, not the NPCs, but the other mice that we did the studies, they get very, very obese, exaggeratedly with this diet, no? And this is chronic diet in mice that are not used to this type of diet, yeah. But definitely, I would say it's obvious that unhealthy diet is definitely damaging for our brain, our heart, our blood vessels. So I would say that I definitely believe that it will be bad.
Are you aware of other neurodegenerative diseases that work in a similar way to NPC1?
So there is many other lysosomal storage diseases that are super similar. They also have Purkinje cells are the cells that are more affected by these lysosomal storage diseases. It's different which is the enzyme that is lost, and so they will be accumulating different components or different molecules inside the lysosomes, but they are very similar. There is very interesting things about NPC. For example, there is they have been discovering a interaction between the lysosome and the mitochondria. And I went to a talk where Parkinson's disease, they were seeing something very similar. And biologically, it's going to be super helpful, no, to understand how this interaction between lysosome and mitochondria are affecting and causing neurodegeneration. So And obviously also microglia, how microglia is involved in Alzheimer, Parkinson, in all the diseases. So you always are contributing to the knowledge of also how neurons die in terms, is similar in some things, no, not everything, but you can still have mechanisms that overlap.
There's a question of detection in humans. How early is this diagnosed generally? Is it?
It can take very long time.
Until...
Yeah, because I think an investigator is already, I don't know if he already make it available for people to use, but I think they wanted to add it as one of the tests that they do to the newborns. So to detect any genetic disease that could be involved. Because, and he found a biomarker in the urine. So I think it's going to, or in the blood, and it's going to be able to be used maybe at birth. But right now, not until the kids begin to have the symptoms is that they begin the process of discovering what it is, no? And there is so many possibilities. One of the ways that the doctors, if the doctor is knowledgeable about the disease, because the disease is a rare disease, is that the kids can have pathology in the liver. The liver becomes bigger, and also the spleen. If the doctor is smart or not smart, or if knows about it, that this is something that happened in MPC, and it see the cognitive or the motor deficit, and then it sees the liver disease or the spleen, then it can get like, oh, maybe this is MPC. So, but for that girls, it took, I think, more than 8 months, something like that, more than a year to discover what was the disease. So, because yeah, they have to go through elimination, like what could be happening. No, it's not that easy.
But as you're finding, it's really important to discover it early.
Yeah, and they are actually, this disease, they had already a trial of a type of a sugar. It's very interesting, a sugar that is going open the lysosome and let that cholesterol to leave the lysosome. And in some way, it delays the disease. No, but the problem of the treatment is that it cannot go through the blood-brain barrier, and it has to be injected directly into the spinal cord, and it's kind of dangerous. And not all the kids are going to have access to that, but because it's a very small population too. They have been able to do the trials and trying to figure it out if it's something that can help.
That's very interesting. So I know that we're all interested to hear a little bit about your backgrounds and how you decided to be a scientist and how you got to where you are.
Yes, I was very excited when I heard that Claude said that he was first generation, because I am a first generation too. And it's funny, when I got into college, I obviously like science. I was a very curious kid, very, very curious. My mom got a surgery, and a gallbladder stone was removed from her, and she brought it in a bag, and I was so curious that I put it in the light, and then the stone get like I don't know, it kind of defrost or something. It got destroyed. And she was like, what did you do? And it was because I wanted to see what it was. So in my mind, I was not familiarized of being a scientist. For me, that you see that in the movies, and it looked like something like being an astronaut, like something that I couldn't never get there, no? But I had health conditions, and I was in the doctors, and I was very little. So I wanted to be an MD, because in my mind, I was thinking that has to do something with discovering and with things like that, no? Like, so I didn't know anything about biomedical sciences or be scientists that do that kind of research. And it's not until I get into college that one of my friends, because we wanted to go to med school, she's like, we have to do research. That's part of being able to go to med school. And she did she was more proactive, and she got an interview, and she then called me and she said, I got this interview, I got this job to do research, and she needs somebody else, and I thought on you. And I was my schedule was already done, and I was going to be tutoring, and I was like, oh, my schedule is already done, I don't have time to go there, it's going to be difficult for me, I don't have time. And then she got very angry, and she's like, oh, you are so ungrateful, I could call anybody, and then I'm calling you and you say no, you should at least go to the interview. So I feel bad, and I said like, okay, I'm going. So, but I'm going to say no because I cannot do it. But then when I got into the interview, Dr. Blanco, that it was the investigator, she treated me like I was accepting the position, and I couldn't say no. I was like, okay, yeah. And she's like, yeah, so you are going to begin this day, so which days you can do it, so this is what we do. And I was like, I felt bad that I was not going to say no, I'm not going to do it, no? So I had to do all the changes at last moment. I did the changes, but thank God that I did it, because once I began to do research, I discovered that that was actually what I wanted to do. And I was so excited with the lab. I was even waking up in the middle of the night thinking that I was still in the lab. I was like, oh my God, I didn't leave the lab. I'm still like, and I was in my home. I was so excited. And after that, I decided that it was definitely what I wanted to do. And I was doing actually frogs. I was working in the retina and dissecting eyes, and it was, and they were doing surgeries. They didn't let me to do the surgeries as an undergrad because I was too young yet. But but then later on, I learned the surgeries to cut the optic nerve. It was fun. I really love it. It was really made me do it.
That's awesome. A couple questions about career. So are there any opportunities or experiences that you would recommend for other first-generation students who are also entering the field of science?
Opportunities. I think definitely I think any student should try to go and do research, no? Like if you love science, and even if you want to do it because of the medical school and like it looks good, it definitely is important, no? Because this is the opportunity that you have to do actually to put in practice what you are actually learning in classes, no? And you get skills, you get also opportunity to work in teams. You also have the opportunity to interact with your professor, to talk about the project, to be able to digest how the science works, no? Like I think something that I didn't know and I learned later is that science is not just what is in the textbook. Science is going on, no? Like we have so much still to discover, new things that have to go and being discovered. We still don't know everything, and we know very little so far. So and I think that's you learn the process of how that goes, how you discover. and I have seen my students now growing and how their skills get better in so many ways, communication and even how they feel about the confidence is very, very important. And I love to interact with them in the lab and talk to them and know how everything is going. So yes, I think, and we have two medical schools. If you ask people in it at Rowan, we of course we don't have that many for everybody, but there are two med schools and there are many professors there, and they want students. And when I did my research, it was actually out of campus. I have to drive like half hour. It was in the Institute of Neurobiology. And I think this is something that I was mentioned to Greg and Shelley before we began, that at that point, if I was you at this moment, and somebody will tell me like, oh, you are going to be a professor in an American university, I would practically laugh and say like, that's impossible. No, because I never thought something like that. I did my undergrad in Puerto Rico, so we speak Spanish there. And I struggled with the English because I didn't have a good English education. So my classes were in Spanish, but my textbook was in English. And that first semester of biology was a nightmare because my exams were departmental. So they were covering 7 chapters per exam, and I had to read a textbook that was in English. It was like obligatory, like the class was not enough. So I struggled so much that first semester. I think I did better in physics and chemistry than in biology. So imagine that somebody goes back and tell me, no, you are going to be a professor in biology and you are going to be teaching in English. I will be like, that's impossible. It's not like, so there are going to be times that you are going to find maybe, or this moment, especially that you are developing, no, you are growing, that you are going to feel that you have obstacles that you cannot overcome, and that's not true. You can. So there are things that can be fixed. There are things that you just need to put the effort. You just need to put the work. It's going to be hard. I'm not going to lie to you, but it's going to be possible. And I think I during this time, and I'm still every day, we have to overcome challenges, and I like that. I like to achieve. I like to accomplish, and I like to try. And don't give up and go for it. And try to grow. No, don't let things to stop you. At the contrary, get that as an opportunity to accomplish something that maybe you think you cannot accomplish, but you will.
Another question that we've gotten a couple times is, when's the time to get started? Is sophomore too early? Is senior too late?
Well, senior, I think is too late. I did it like in my third year because I did 5 years, but I think, yeah, I will begin as a sophomore, and I think like the first year I will be trying to figure it out which labs are around and what is the science that I would like to do or something. But yeah, sophomore will be ideal, and I think it's great when I got students. I have students from TBS and they begin as a sophomores, and when they get to senior, at the senior point, they are like grad students in my lab. And I see them and I'm like so surprised, like how independent they are, how good they are, and like it's really amazing. No, they get to that point that you see like they are almost graduate students. So yes, sophomore I think is the best. I think sometimes, freshman you are beginning and maybe you should concentrate in your classes and make sure that you set up with the routine of the university and what it is, and then go for, at least that's my opinion, but not necessarily has to be like that. No, but I think that seniors they are good. Sometimes they have skills that they have get in their courses, and I really see also that they get the techniques faster than even the younger. So
that's great. That's one thing that I noticed in your acknowledgement slide is you have students from a number of different departments. You're in the MCB department here at Rowan, but you've had students in the bio department and other departments.
yes, definitely. The majority of my students, I think, are from bio, no? And I have had from biochemistry, TBS, MCB, yeah, definitely. Any student that is interested in doing biomedical science, no? I know that I would like to have more. I think it's funny because before the pandemic happened, it was the first time that I got a lot of students, because one of the reasons is that I was getting funding. I was like, now I have funding, so now we have more to do. So, and I got all these big groups, the picture that we are together with the all with the all. It was the first time that I think I got like 17 students. I never had that many, but then the pandemic happened. It's not that easy for me to get too many students because I like to train the students myself and to be able to check on them. And especially that time I did it too because I have all my seniors that work with me for many years and they were going to help me to train the new ones. And then the pandemic ruined everything, but it's okay. We are still, so let's see if when we come back, I don't know how many I will be able to recruit because it depends how everything. I'm putting all the weight in this summer, how the summer goes. I already have my students for the summer, and if they are trained well and they are ready, maybe I can get more in the fall.
Great. I think maybe one last question. How was moving from biology or more general biology to neuroscience for you? How was that transition? And was that scary at all, or was that natural?
It was, yeah, it just happened by chance. I think many people, they finish in their fields because that was the field when they began by accident. No, I got that interview just because somebody told me. I was not trying to go into neuroscience purposely. So that's how it happened. I think I never thought I'm going to for neuroscience. I was not doing neuroscience in my mind. I think I didn't even know about neuroscience. No, like I was sitting in the seminars there, and they were talking about action potentials, and it was like, and especially in the institute, they were speaking in English, so I was not understanding anything. It was like seeing graph there that I was like, what are they talking about? But yeah, with time, it went. It just felt natural, no? Like I went from studying the retina to then study glaucoma in mice, and then to go from mice to Alzheimer's disease from glaucoma, and then now MPC. So you just stay and go in the same field. That's how it happened.
It just happens naturally.
Yeah, it just happened. You just go now I'm doing development, and I never did development before. No, you follow what the science is giving you, all the opportunities that are opening. No,
and where your interests are.
Exactly. And the thing with neuroscience is that if you see there is physiology, like it's biophysics, or there is cell molecular, like it actually have all the other sciences on it. So one time I actually was studying a diabetic mouse model, and we were studying how diabetes was affecting glaucoma, but then I noticed that they were urinating so much and they were so skinny, and so I got the liver, and I did staining in the liver, and I found the liver was larger. I found changes in the liver, and I put it in the paper. So I think once you know cell biology, I'm actually a cell biologist and neuroscience because I'm working in the brain, and I'm working in the, but I'm mainly when I'm in between neuroscientists, the majority of them do electrophysiology, and I don't, so I feel like I'm not a neuroscientist. But anyway, but it's just because I'm working in the brain, no? So yeah, so that's something good about neuroscience. You can do any type of science in neuroscience. So
that's wonderful. Thank you so much for the time that you spent with us today, and for all the information. There was close to 50 people here, as far as I can tell today, so that's wonderful.
Yeah.
I want to remind everyone that's still here, everyone, well, you guys, that our next seminar will be with Dr. Patrick Herman. And he is in both the Department of Environmental Science and Biological Sciences. And that'll be Monday, not a Tuesday, April 12th. It will be at 3:30, not at 11, the way these couple have been. But we look forward to seeing you guys there. I hope that it seems like everyone's enjoying this, so that's great. I hope that you guys enjoy this and you get something from it, and that something can be just one little thing that sticks with you. Okay, maybe it was like 6 little things for me, but thank you again, Dr. Soto-Reyes, for joining us.
Thank you so much, everybody, and thank you for your questions. And if you have any questions, email me or anything. I'm not an MD doctor. I cannot answer questions about diseases, but I can answer what the research is going on, what we are getting into research. This is something that I discuss with my students all the time. What is coming? What are the goals in science? How we want to cure these diseases? So that's the hope. No, that's the good thing about science.
It's great. Dr. Thomas, do you have anything to add? All right, with that, thank you all again for joining us, and we hope to see you again. I guess that's next week, next Monday.
Bye-bye. Thank you.
All right, and that leaves just us. And I will stop recording as well. I'll try to stop recording. There we go.